Howdy guys. Okay, this is video 1 in the series, but if you were in class on Friday, don't start here. You don't need to sit through class again. You're going to be bored to tears. Instead, go to video 3. 3 picks up with the new information. So if I go to my to-do list, this is video 1, been there, done that. This will be video 2. This is the new stuff. So what I want you to watch, if you were in class on Friday, 3 and 4. If you were not in class on Friday, or if you were confused on Friday, yeah, start here. You'll get all the information and learn it well, okay? So you got a bit more to do to make up for being absent. Not bad. So I'm going to keep it painless, though. All right, so what the goal of today is to get from DNA to actually traits to you. So you get blue eye gene from your parents. How does that actually give you blue eyes? So we're going to go through a process to turn that DNA into proteins, and then the proteins will then create your traits. That is what's called gene expression. It's using the information in DNA. We're going to need 3 substances: DNA, RNA, and proteins. So by the way, we're finally on number 1 in the guided notes. The DNA is the information. The RNA conveys that information from the nucleus out into the cell. And inside the cell, the proteins are the workers that they're then going to use the information to make you. It's a very, very nice system. All of this is accomplished in 3 steps. Those steps are abbreviated in what's called the central dogma, meaning the most important idea, the critical idea of biology. The central dogma is DNA, arrow, RNA, arrow, protein. Memorize. Guarantee you that's a question on the next test, is what is the central dogma? You can write it like this. That is a perfectly acceptable answer that gets you all the points. Or you can write it in English. In English, it is simply DNA is used to make RNA. That RNA is used to make proteins. Right? So it's a great process. I enjoy it. This was one of my favorite areas when I was a fresh person. So hopefully you will too. This is how the book abbreviates it. Represents it. This is a metaphor the book uses. The DNA is a master plan. The master plan is kept in the offices where it can be protected. But copies of the master plan can be made. The copies are the RNA. Those copies are sent out into the world where various workers, proteins, turn that into actuality. They're the ones that are going to actually build the building. So it's a good analogy. It makes sense. Hopefully it helps it stick in your head a little bit better. Now to carry this out, we're going to do 2 steps. Step 1 is transcription. Step 2 is translation. Transcription is going to turn DNA into RNA. Okay, so this is the very first part of the central dogma. Translation takes that same RNA and then turns it into protein. So we're just cracking the central dogma into half. Each half gets its own process. Now, these words are pretty familiar. So to help you tell the difference, let's do a little bit more detail on each step. Transcription is all about copies. Think of a transcript. A transcript is a copy of your grades. We take that transcript and we send it out to all the various universities you apply to. But they all get the same copy. So transcription takes DNA and creates an mRNA copy. Because if you look, this and that are pretty close. There's some differences that we will talk about. And then this copy is sent out into the cell. So transcription just creates an RNA copy. Translation does not copy. Translation changes the language. Okay? So we're going to go from RNA language to protein language. And if you notice, these are not the same looking. So in video 3, we'll talk about how we translate from this language to that language. It's like going from English to Latin. You go, I'm using letters, but I am not using them in the same way. So video 3 is going to be a bit more complicated, but you can do it. All right. There's two things I want to review before we actually go on to processes. The first is the difference between DNA and RNA, and the second is going to be types of RNA. Now, you knew the difference at one point in your life. You might remember it, I hope so. If so, this will be easy. If not, well, buckle in. There's 3 differences you need to know. The first is the number of strands. DNA is double-stranded. RNA has just 1. So DNA is 1, 2 strands. RNA, very lonely, 1. Right? So pretty obvious difference there. It's big. Second one involves the sugar. So if you notice, the sugars are pretty similar between the two, but in DNA we have deleted an oxygen. That oxygen is gone. So we call it deoxy, for delete an oxygen, ribose, which is the name of the sugar down here. Now, this sugar over here has all the oxygen, so it is simply ribose. Same sugar, only difference, 1 oxygen. The D in deoxyribose is where the D in DNA comes from. The R in ribose is where the R in RNA comes from. So, important. Small, but important. Finally, bases. 3 bases are going to be the same. A, C, and G. A, C, G are in both. A, C, G, A, C, G. The important difference is DNA has thymine, we learned about that, but in RNA it's replaced by uracil. So T replaced by U. So 1 base change, which means over here we say A pairs with T, now A pairs with U. We can handle that. Okay, so that's the 3 differences. This figure sums them up. Strands, 1 versus 2. Ribose versus deoxyribose. Thymine versus uracil. Memorize, okay? You had it in your head once. It's easier to relearn something. Last thing I want to talk about is the 3 types of RNA. That's here, mRNA, rRNA, tRNA. These are the abbreviations. So in your notes, I'm going to ask for abbreviation, name, and function, right? So if you want to go ahead and fill in the abbreviations right now, this is number 6. You can do so. We're going to go one by one. I'll talk about each one individually with pretty pictures. Start with the most important, which absolutely is mRNA. This is the one we will spend the most time on. Its name is messenger RNA. Its function is it carries genetic information out of the nucleus. It does what a messenger does. A messenger says, all right, I pick up my information here from the DNA and I carry it out to the cytoplasm. I go from point A to point B. Okay? It even looks like a sentence, which messages are filled with sentences. It's a long string. Right? Most important, and the one we will deal with a lot. The one we will never deal with ever again is the weird one. rRNA stands for ribosomal RNA. So here's the name. Ribosomal RNA is in the ribosome. And if you remember right, what do ribosomes do? Ribosomes make proteins. So rRNA helps the ribosome make protein. How does it do that? We don't know. We know it's there. We know if we remove it, the cell dies because it can't make proteins. But ribosomes are small and the process is fast. So there's lots of theories about what rRNA does, but it's all theories. It's all questions. So I'm going to say it exists, and we're never going to talk about it ever again. We don't know. Too confusing. Okay. The third will come back. We will talk about this. They're called tRNAs. T technically stands for transfer. I call it translator because that makes sense. You can call it whichever one you prefer. I will accept transfer or translator, whichever one fits in your head. And it does what a translator does. It looks at the DNA and says, oh, not the DNA. It looks at the RNA and says, oh, the RNA says ACC, then I need to add this to my growing protein. So, like, for example, if I wanted to translate from English to Spanish, I would look at the English word and go casa. That wasn't English. House. Okay, then in Spanish, I need to write house. So it does that. It translates from one language to the other. Alright guys, you survived the first video. Not bad. I am going to stop here. This will be video 2. So go ahead and fast forward to that one and let's see what happens. See you there.